everyone. Welcome to this new episode of our Student Spotlight series, where we're able to showcase some of the work our past students have done. I'm Charlotte. I'm one of the outreach coordinators here at CCIR, and it's our pleasure today to have with us Chris, who did one of our courses in the Future Scholars program. So to start off with, can you just introduce yourself? So tell us a little bit about who you are, where you're from, and what year you are in school.
My name is Jihoon Ryu, and my English name is Chris. I live at Korea, so I study overseas in an international school at Daegu. Daegu is one of the cities in South Korea, and I'm a sophomore.
Great. And then can you tell us a little bit about the course that you did? So what the course was, if you remember who the mentor was, and just in general what it was about.
The course was about computational biology, and my mentor was Dr. Murray. And the course obviously is computational biology, but it was very interesting for me to delve into another sub class of biology as technology is rapidly increasing. More techniques and coding languages are coming out in the internet for available for even students like us to access it. So yeah, I took computational biology and yeah.
And then did you have any experience with this subject in school or in a different platform before you did this course?
Before this course, I also took another summer camp last year, and it was called MIRCOR, and I learned also about computational biology there, but it was not it. There were like tons of more students, like about 40, 50-ish students there compared to this scholar program where only 5 students go against one mentor. So I learned about computational biology, but not like this deep. And as you know, the mentor cannot focus on 40 individual students, I couldn't ask much questions or like actually connect to the topic. But I with this program, I was able to learn more and delve deeper into this subject. And also, this program utilizes and persuades students to use the MATLAB coding. But instead of MATLAB, we used the R program, which is known as easier for students to access. But still, MATLAB was very interesting and it was way advanced, and it was a better platform to visualize data and graphing.
Great. And then just for any people tuning in that might not understand what computational biology is, can you give a summary, just kind of in general lay terms, about kind of what computational biology is?
So human has a genome, and genome refers to the entire genetic data of the body, and the DNA is really, really long and is made up of small pieces called nucleotides, and a human genome can have thousands, millions of them. And computational biology, however, just can analyze that thousands of nucleotides by using simple codes and by using for loops. By using loops, you could just analyze an entire genome. For my case, I analyzed a bunch of data sets for yeast, and instead of handwriting, which was a method that was used a long time ago, right now, like DNA sequencing models and CRISPR technology has also been in our site recently, and yeah, it has been just advancing throughout time, and now it is taught more to students.
Great. And then to shift into your specific research project, can you tell us what your research project was and how you decided on that topic?
So my research topic was about antisense transcription, and transcription is a term that means where RNA is a small nucleic acid that is produced from the DNA, but antisense is when this is the gene. Antisense is the opposite strand, and the opposite strand also produces that RNA, but it was first discovered in 1990, which was extremely recent, and scientists didn't know what it did, so they even called it a nature's trash. But right now we are certain that about like 72% of human genome are that RNA. So therefore, there's gonna be a reason if we have so many of that. And in my results, I found that it was a crucial regulator of gene expression, and gene expression results refers to how many protein are formed for our body. So our body may need a lot of protein or a little bit of protein to do basic body functions. So antisense transcription can transcription can impact how many or how less proteins are formed. And the reason I chose this topic was antisense transcription. I first heard that term in this course, and it was extremely interesting because unlike other science concepts, it was discovered in 1990, which was like 3 decades ago. So the researching process was very new, and I had the opportunity to read also the mentors articles on online, Miss Dr. Murray's papers on online, so it was very interesting.
Great. And then can you tell us about your research methods and process? So what type of research did you conduct and how was the process of finding data?
So first of all, I tried to grasp the basic knowledge of antisense transcription, how it works, and Dr. Murray facilitated me through that process as he's very efficient in teaching us the mechanisms. So, but after learning about basic transcription and what happens in the body, I explored about antisense transcription by referring to a lot of papers, and there are a lot of very good videos on YouTube because I'm a visual learner, so it was better for me to watch. For example, like I don't know what microRNA means, so it was better for me to watch like what it was and how it was formed through drawings and diagrams. So YouTube was definitely a helper for me. And then aside from that, I read a lot of articles. Mainly they're abstracts because I couldn't go through every single word in that paper, obviously. And yeah.
Great. And then did you discover any interesting findings through your research? And can you tell us what you think the importance of your findings are to the field or to the general public?
So I found out that so transcription factors are what helps the RNA polymerase, which is a protein that helps produce the RNA. So antisense transcription had the trend of transcription factors attaching to it, and maybe that's the reason why it is a regulator of gene expression, because if more proteins bind on that, other proteins cannot come and bind, so therefore it creates competition. And this knowledge was already known in the public, but still it was a new experience for me, and it was also interesting to see that some of the results that I produced didn't match other papers. For example, so the RNA produced in the antisense strand is known as unstable because it degrades fast and it just goes away. But in my results, it came out that antisense strand was more stable than sense strand. But I checked with my TA about that, but still the results still came out okay. And the reason would be that sense transcription levels were so high that it just flawed the results. And for the general public, maybe a lot of medical concepts relating to the antisense transcription came out into the public. for example, AS oligonucleotides, I think, and other medicine that came out due to the discovery of antisense transcription. And recently, I believe that like the U.S. Food and Health Department has accepted two types of medicine from antisense transcription to treat disorders. Yep.
That's very interesting. Do you know what those disorders are by chance?
I wrote that in my conclusion section. So the two are is U.S. and Food and Drug Administration accepted two antisense mediated therapies for the treatment of Duchenne muscular dystrophy and spinal muscular atrophy.
Interesting. And then have you had any experience or have you started the publication process, or is that something you're thinking of doing?
I almost finished writing my paper. I am currently in the revising and the editing process. So before the end of the week, I am very excited to submit it to the CCIR evaluation so I could get a evaluation report of where I could potentially submit it to. And if I get a matching journal that I am really interested in, I will be publishing it.
Right. And then now we'll shift into just your general experience with CCIR and with the Future Scholars Program. So can you just give us a summary about what this experience was like for you, especially doing a high-level research program and research project like this as a high school student?
So, in South Korea, not much people are studying computational biology as is overseas, and not much people are in like the genetic field specifically. So therefore, as a student also, it was a very new experience. And for the meetings, I found the TA sessions very helpful because I was able to prove my codes, and if the codes are flawed, then that means my results will be flawed. So my TA would be a checker for that, and she helped create a foundation for my research. So I really like how there are TA sessions, and I also love that our mentor was actually really experienced with this subject and is actually a publisher, a really famous book writer, and also teaching at a university, which is very cool.
Yeah, that's great. And then since you mentioned the staff of your program, so the mentor and then also your TA, we in these programs, we really like to emphasize the aspect of mentorship, as you probably know. So we like to have our mentors be more of guiding students on their own learning process rather than the traditional teacher role where they teach you and you kind of let that sink in and then regurgitate the information in a test, for example. So can you tell me your experience with your mentor and your TA as well, as you moved through the first half teaching course or first half of the course that focused on teaching and then into the latter research half of the course and how they helped guide you through that process?
So in the first half, as you say, it was just a teaching. So it was mostly basic functions of our body. So like transcription, translation, DNA replication, which are the three most important processes in our body to produce proteins. And at the second half, Dr. Murray was more focused on each of the project choices because he gave us 3 project choices and the associating data sets for each project. And he explained each project very in detail, like in a detail that we can't even find really on the internet. And like the exact numbers were also very interesting for me to find. And the TA was very the TA. So we had to solve a worksheet for every week, and the worksheet was about coding in MATLAB and analyzing genetic data. So our TA did an excellent job of guiding us through the worksheet solving process. And it was the knowledge that was put onto the worksheet was put onto my research paper. and it was very helpful because all that worksheet process, it might seem tiring, but all that built up for my knowledge to write the research paper and how to produce my own results based on the data sets given.
Great. And then how was this research process and the course in general, how was that experience for you compared to papers or projects or courses that you've taken in high school?
So, this is my first time writing a research paper on biology, but however, I have written a lot of academic essays, but however, this time it was different because I had to limit my vocabulary in biological terms. I couldn't really express my own opinions. I had to stay objective to the results because it's going to be published, and it was a new experience. So I had to search ton on Google about how to actually write a research paper. And the mentor and the TA actually devoted one class for explaining about how to write a research paper, which was very helpful. I didn't know anything about the discussion section or how to write an abstract or how to write a methodology section. So those sessions were very important to me in writing the research paper.
Great. And then just for those listening to clarify, so our Future Scholars program is our group course. So each course has a maximum of 5 students per course. So can you tell us a bit about how it was working with other students, if you remember like where they were from and what that experience was like for you?
Most of the students were from America, I believe, and I think that working with them was a little bit better because if I was alone, I would have gotten more pressure in answering the questions of the worksheet, and therefore my peers would help out with answering those questions. And when when I had one peer that had the same topic as me, so whenever she asks a question, I listen thoroughly to see if it helps for me too, and I believe that my questions also helped her too. So maybe it was like it was even it wasn't spoken out. It could have been like a telepathic question asking, like helping each other. And I hope it was beneficial for all of my peers because it was beneficial for me to work with my peers too.
That's great. And then just a few last questions. Do you think this course has given you any insights into your future? Has it informed or shaped how you think of your path moving forward?
So I want to still study about biology in my university years and also go into my medical side of my hoping career. So I believe that this is my first research paper, and my first research paper, even though the topic was extremely complex, as as much as it's complex, I think it would serve as strong as basic foundation for me, and it was a crucial step for me to step towards writing a better research paper on any kind of topic in general, and especially biology. And I came upon this important code, MATLAB, and I wasn't aware of this coding language before even taking this course. So I think I will be utilizing this code much more in writing my research papers. And also, the things I learned on biology about DNA, RNA, and all that stuff has been strengthened in this course because I was not sure about multiple concepts, even in the basic sides of biology. But now my basic knowledge of biology is structured and is very strengthened by my mentor and the TA. I think it'll be better for me to like grasp more complex topics that I will be learning in the future. So I think it'll be beneficial.
That's great. And then the last question for you, what would you say to someone who asked you what the value of doing a project or a program like this is, especially for a high school student?
For a high school student, writing a research paper is not really a common experience in the field because that is known as what researchers and scientists do. So I think the process is extremely fun because you feel like a scientist. You actually feel like a researcher when you are learning about computational biology, because that's what I took, and I hope it's for all the other subjects too. And becoming a researcher is very, like, you feel proud of yourself because as you put all the equations in the research paper, and I made up one of my own equations too. It was a new experience for me, and for people that are hoping to write more research papers or are going to go to a career that is related with the topic that you are going to learn in the CCIR Scholar, it will be extremely beneficial for you to listen to your mentor and TA or make a mutual connection with them so that you can learn and get the best out of it. And most importantly, you shouldn't be scared of the process because it was my first time, and I think I pretty got through it very well because you have the most experienced mentor and the most experienced TA by your side. So don't be afraid on researching and asking questions.
I think that's great advice. Thanks for that. So that's about it. Thanks so much for chatting with me, and congratulations again on your nomination to our Spotlight series.
Thank you.